Okay, sorry for being a little bit late. I actually needed a little bit more than 2 minutes to finish the Zoom recording. I thought 20 minutes would be enough, and I gave it 20 minutes, but it needed a little bit more time. So it was at 98% when I wanted to start class, and I decided rather than risk fouling up the Zoom recording, that I would start class just a little bit late. All right. Are there any questions? If not, let me remind you we're going to spend a little bit of time on the lecture. And we are behind on chapter 1 because I'm supposed to finish today, and there's no way I'm going to finish. And then we're going to do lab 1. So, there's only one date in this lesson that you do need to know, and that's this first one in this slide here, and that is greater than 3.5 billion years ago, ancestors of bacteria were seen in the fossil record as the first life on Earth. And here you can actually see some actual fossil bacteria,
Are
and
you
this
sharing
is
your
an artist's
screen?
rendition. I'm sorry?
Are you sharing your screen?
I'm not sharing my screen. I thought I was. Just a minute. No, I'm not. My apologies. Make sure I'm recording. Yeah, I'm recording. All right. So, over 3.5 billion years ago, there's the ancestors of the first bacteria that were seen on Earth as a fossil. And that was the first event, you could say, in the history of microbiology. Although this is a little bit before microbiology, but and that's the only date you need to know. I will refer to all these others with a date, but you don't need to know the date. The next major advance in microbiology was in 4,300 years BC. The Babylonians wrote clay tablets recording their beer recipes. And they didn't know it, the Babylonians, but the reason why they were able to get beer was because of microorganisms fermenting whatever it was you make beer out of. I think it's malt or grain or something like that. And then the next one would be 600 AD. Like I said, you don't need to know the dates. Is that the Mayans made a fermented beverage from cacao that we call today chocolate. All right? And generally speaking, you don't need to know the dates, but you should know that before the Mayans were able to make a fermented beverage using microorganisms to make the fermented beverage that we call chocolate, there had to be the Babylonians who wrote their clay tablets about making beer, and they're using microorganisms to make the beer. They didn't know that, but they were using microorganisms to make the beer. And then before that, there had to be the fossil record of the first life on Earth. So you should know the relative order of these things. Any question about that? But you do not need to know the dates except for this first date. The next major advance was in 1590, and Janssen developed the compound microscope. Obviously, you needed the invention of the microscope before you could have microbiology. However, microscopes were not used on living things until 1665, and you do not need to know the year, when Robert Hooke reported that he found that living things, mainly plants, were composed of little boxes that he called cells. And let me see if I can even do this. I'm going to try and explain why he called the things that he was looking under the microscope cells. When he looked at them, hopefully this will work. I can't draw very straight, but you're getting the picture. He saw cells like this, and it reminded him of the cells in a monastery where the monks lived in the monastery. So they had a little cell on a hallway, so this would be the hallway in the monastery, and there were these little rooms where the monks lived, and the monks called those cells. And Robert Hooke was reminded of those, and so when he saw cells under the microscope in plant cells, he too called them cells. Any question about that? That marked the beginning of the cell theory, where the theory was that all living things were composed of cells. This theory was further developed and officially written out by Virchow, but that was much later. Initially, microbiologists or biologists only saw cells in plants, and that was because the microscopes back around 1665 and Robert Hooke's days were too primitive to see cells in animals. And why would a cell of a plant be much more easily seen under a primitive microscope than a cell in an animal? Come on, you guys should know this. You took the preliminary class. I'm not moving forward until somebody answers, even if it's wrong.
Can you repeat the question, please?
Why did the early microbiologists not see a cell in an animal, but they were able to see cells in a plant?
Because they were like leaves are transparent sometimes.
That's true, but no, that wasn't the reason. Because you could get a thin section of a tissue from an animal. What's different about the cells of a plant and the cells of an animal?
Does it have to do with the cell wall?
Yeah, plants have a cell wall, something that's very thick and rigid, and it's very easy to see under the microscope. Animals do not have cell walls, and because they didn't have a cell wall, the primitive microscopes weren't very good, and so they could not see the cells in an animal because all they had was the cell membrane, and it wasn't as easily seen as the cell wall. The above is not specifically microbiology, but it was events that were important and had to happen before microbiology could happen. A history of microbiology largely concerned two hypotheses about how life originated, and these two hypotheses were competing with each other to explain how life began. Some people believe that life arose by spontaneous generation, where life arose from, the elements, after a vital force came in, and then life could arise. Others said, no, life can only arise from another living organism. And they gave an example for biogenesis, and that is you never got a baby unless you had the parents around. Okay? Anyways, microbiologists argued about these two theories for many years. It included things that were logical but not terribly scientific. And that is when you took a log and threw it into the fire, sometimes you could see a newt crawling out of the fire. And the spontaneous generation people would say, aha, there's evidence of spontaneous generation. You put wood in fire and you spontaneously generated a newt. Another one would be where flies would emerge from the manure. And I don't know what the elements were getting together, obviously the manure and something else, and then the flies would spontaneously generate. And then another one was where maggots would arise from a decaying corpse. And they all said this was evidence of spontaneous generation. None of them were scientific. Some were amusing, such as somebody printed out a document. If you put sweaty underwear in a jar and you put wheat in the jar, it would spontaneously give rise to mice. In reality, the mice got into the jar to eat the wheat, and then because it was in a jar, the mice were unable to get out of the jar. We're going to skip that. Skip that. The first evidence for or against spontaneous generation came from Francesco Redi. And he's also important because not only did he add to the debate about spontaneous generation, but he's also the first person to write up and document a controlled scientific experiment. So he's actually very important in science. What he did was he got, I should say, the first in biology. He might not have been the first in science. What he did was he got 3 jars and he left them open to the air. And then he got 3 jars and he completely sealed them. And he noticed that maggots appeared in the open jars and there were no maggots in the sealed jar. And he said, this is evidence that spontaneous generation does not occur. And the people who believed in spontaneous generation said, no, 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 you sealed the jar. The vital force wasn't able to get in and then give rise to the maggots. So what he did was he made 3 more jars and he covered them with a fine wire mesh. And he noticed that there were no maggots in these 3 jars open to the air where the vital force could come in. And so this was the first evidence that spontaneous generation did not occur. And generally with larger organisms, people after Francesco Redi's day did not believe that a large organism like a maggot, which isn't that large, but you get my picture, it's something we can see without the microscope, that people came to the conclusion that organisms like maggots could not spontaneously generate. However, the microbiologists who believed in spontaneous generation still believed that microorganisms, which were much smaller and simpler than a maggot, could spontaneous generate. And what they would do is they would take a container and leave it open to the environment, put sterile media in it, and then microorganisms would grow in it. And they would say that's evidence of spontaneous generation. Of course, the microbes were in the air and in the tube, but the people didn't know that. And so let's go on. While this argument about spontaneous generation or biogenesis were being argued by the early microbiologists, we had the first observations made in microbiology. And that was done by Antony van Leeuwenhoek, who was the best lens grinder and microscope builder of his day. And he did his studies from 1673 to 1723, which is I think when he died. And he used his microscopes to describe all sorts of microorganisms in his teeth scrapings, in microorganisms growing in rainwater, in peppercorn infusions, and then microorganisms in the River Thames, the river which supplied the city of London with water. And believe me, the people of London weren't too happy to learn that there were microorganisms in their water supply. Here is his early microscopes, and to our day this is very primitive. You'll notice that this is a very simple microscope. It only has one lens, which is acting as a super duper magnifying glass. But he was the best lens grinder, meaning he actually made his own lenses by grinding them down, and then built his microscope. And you can see this pin, that's where you stuck something to observe. And he was such a good microbiologist and built such a good microscope that he could see these cells over here, which were cells so small that they were not seen again for about 100 years. And if you don't know what these are, he saw and then recorded in his drawings pictures of bacteria. And they weren't seen for 100 years because the other microscope builders in his day could not see them because their microscopes were not good enough to see them. And it wasn't until 100 years later that the microscopes got better that we could see bacteria. But these are actually bacteria that Antony van Leeuwenhoek saw. All right, let me finish this, and that is Rudolf Virchow proposed that cells could only arise from pre-existing living cells. So he actually wrote it up, the theory of biogenesis. And then the theory of biogenesis was finally settled by Louis Pasteur, who what he did was he disproved the theory of spontaneous generation and therefore confirmed the theory of biogenesis. What Pasteur did, he did many things in microbiology, but for this argument, he discovered that the air was loaded with microorganisms, and that microorganisms could fall out of the air and then contaminate the media. And he also learned that microbes could be destroyed by heat. And so you could sterilize the media simply by heating it. And what he did was he made his ingenious swan neck flask experiment to disprove the theory of spontaneous generation. He got a flask like this, and then he put the media in it, and then he heated the flask to bend it in a swan neck, okay, which is this shape here. And then he sterilized the media by heating it and then allowed it to cool. What he saw was there was no microbes growing in the media, even though the vital forces could come from the air and go up the tube and go into the media. He found that if he took the media and he brought it down to here and then brought it back, that microorganisms would grow in the media. And what he did was he showed that there were microbes in the air and they could fall into the tube, but they would settle here because by gravity they would not go up the tube, this part of the tube, and then contaminate the media. And by his experiment, we finally proved that spontaneous generation of microorganisms did not occur. Any question about that? All right, actually I've got two here already, so I'm just going to stop the lecture here and then continue with the lab. And we are behind schedule. I need to get to, let me see here. So what slide is that? Slide 52. I need to move to slide something like 67. Yep, I need to get to slide 67. After slide 67, I can stop, and we will not discuss slide 68 through 78. But I do need to get to slide 67. All right, any questions? I don't know why I need to save that twice. Oh, there it went. All right. If there's no questions, let's go on to the lab, lab 1. I think this is the one I want. I've got a teacher version and I got to put a T on it to let me know that's the one I want. And then I've got another one which is the lab that I post, meaning it's not highlighted. Nope, this is the wrong one. Let me take a look at that one. Yeah, this is the one I want. All right, you should know the learning objectives. Know that microorganisms are almost everywhere. Describe the purpose of culture media. Describe what colony abundance is, colony diversity, and colony morphology. And then be able to define the terms. Any question about that? I suppose I should mention you should do some reading before you do the lab. Microorganisms are organisms that are too small to be seen with the naked eye, and that does include viruses and prions, which are not really cellular organisms, and it does include helminths or worms, which are not really microscopic. Okay? The phrase ubiquity of microorganism refers to the concept that microorganisms are essentially almost everywhere. In this lab, we're going to investigate the abundance and diversity of microorganisms, and we're also going to learn basic lab protocols, including the use of solid media, labeling cultures, procedures for disinfecting lab benches, and procedures for washing our hands. A culture medium, also known as a growth medium, is a sterile laboratory preparation that contains a variety of nutrients that are required for growing microorganisms. And we'll talk about how we sterilize it in a later, I think it's in the lecture, later lecture or lab, where how we sterilize it is we put the medium in an autoclave. And if you don't know what an autoclave is, it's a giant pressure cooker. The culture media may be liquid. These we call broths, and they're commonly put in a test tube. And we can add agar to the liquid medium to result in a solid medium. We have a video for you to watch on how to prepare a pour plate. And you should note how the culture medium needs to be sterilized prior to its use for pouring it into the plate. when we have a solid media, we can grow it in a plate like this, and this is usually called the petri dish or a plate. But we can also have solid media in a tube, and this is called the deep, where the media is just flat across, and that's a liquid broth. But we can pour the media containing agar when it's still liquid, and then tilt the tube at an angle to get the agar to form a shape like that, and this we call a slant. Any question about any of that? All right, there are several different types of media. In today's lab, we're only going to learn about general purpose media. It's a non-selective media that is usually complex in composition, and it's used to support the growth of many different organisms, but not fastidious organisms, by using peptone, yeast, and beef extracts. A fastidious organism is one that requires added ingredients, or else it's not going to grow. So a fastidious organism is one that is fussy. Neisseria, gonorrhea, and what else? Streptococcus pyogenes, which causes strep throat, is a fastidious organism, and they will not grow on a general purpose media. 3 general purpose media that you should learn are, I'm going to start down here because this is the simplest one, nutrient agar, or Na, and then you can take the nutrient agar and add yeast extracts to it, and we call that nutrient agar with yeast extract, and that's usually abbreviated Na. And then you can take tryptic soy agar, another general purpose media, and that we call TSA. The reason why microbiologists use general purpose media so frequently is because they grow a lot of organisms and they are cheap. They're also fairly easy to make. Okay, so these are the media that we use most of the time in microbiology. Any question about any of that? We will talk about other media in a later lab, but for now we're only talking about general purpose media. Everything in the lab should be labeled, and it's vital to label the petri dish or the tube correctly. So when you label something, you should always include your name or your initials, the date, the type of media, and the inoculum. The inoculum is what you're growing. The incubation temperature is optional. You may take record the incubation temperature as well. So this is showing you an example of a Petri plate labeled with the person's name, Travis, what they're growing, E. coli, that would be the inoculum, the date, the media, TSA, and then there's the temperature that it's grown at. When you're labeling an agar plate, you should label around the perimeter of the base of the plate, meaning the bottom of the plate. You don't want to label here because it will interfere with your seeing or observing the growth on the plate. And why you label on the bottom of the plate is that if the lid gets separated from the bottom, the cells will always be on the bottom of the plate. And I once had a person I was working with, she had like over 300 plates and we're carrying them on a big tray from one location to another, and she tripped and she spilled all the plates, and all the lids got separated from the bottoms, and she had wrote all of her information on the lids. So all of those plates had to be thrown away because nobody knew what they were. Any question about that? Meaning nobody knew which lid went with which bottom. So we write on the bottom. Another reason for writing on the bottom, I guess I can mention that, and I don't know how visible this will be, is that here's our Petri plate. Obviously I'm pretending. When we put it in an incubator, we don't put it in right side up. We turn it over and then put it in upside down. And I think I've got a discussion about that someplace down here. And that's because when we put the plate in the incubator, the warmth of the incubator will make water come out of the agar, and then that agar will condense on the, boy, I can't get that to show, will condense on the, there we go, will condense on the bottom of the lid, and it'll form droplets which will drop down into the plate, dispersing the bacteria. So we put things in the incubator upside down, and then the agar is on the top, and if moisture comes out of the agar, it will condense on the agar. The agar will then wick the moisture throughout the agar, so there will be no drops forming, and even if drops did form, they would drop on the lid, and there's no growth on the lid, so we don't care if something were to drop on the lid, but it won't. So that's why we put things in the incubator upside down. And if you write on the bottom, you can more easily see it when it's upside down in the incubator. Any question about any of that? All right, some terms you should know. The inoculation is when you're physically putting the cells into the medium. The incubation is when you then put the tube or petri dish someplace for it to grow. And usually we put the stuff in what we call an incubator, a special box that is a controlled environment for growing microorganisms. Usually incubators at the very minimum will add a little bit of humidity and will warm up the environment to encourage microbial growth. But complex incubators can do other things, like put in different amounts of oxygen and carbon dioxide. All right, I talked about that. When we look at the bacteria colonies that grow on a plate, a solid plate, meaning solid agar, there will be differences. And you can look at those differences to compare what is a fungal colony from a bacterial colony. I've got a little picture of that here. Actually, that's a picture of an incubator. Somebody's growing cells in it. And there's a video you should watch on binary fission, how most microorganisms grow. But a bacteria colony, let me blow that up a little bit, is distinct and has a very smooth edge. And so we can see that these are bacteria colonies. And then these that have an indistinct edge and they look fuzzy or hairy, these are fungal colonies. Any question about any of that? So when you look, you can frequently distinguish between a bacteria colony and a fungal colony. That doesn't apply to 100% of the cases, but it does apply to 95% of the bacterial colonies and 95% of the fungal colonies. All right, where was I? We already mentioned that. You should know that a colony is made up of tens of millions of cells to perhaps hundreds of millions of cells. All of them may be derived from one or very few cells. So when we look at this colony, it may have been derived from one cell or very few cells. Any question about any of that? When we look at the colonies, we can see different colony characteristics, and you can see that the colonies have a shape. There can be circular colonies, rhizoid colonies, irregular colonies, filamentous colonies. And the edge of the colony we call a margin. It can be entire, which is what a circular colony has. It can be undulate, lobate, curled, rhizoid, or filamentous. If ever I ask you to differentiate, I will ask you to differentiate only between entire and these three and these two. Meaning I'm not going to ask you to differentiate, is this colony got a margin which is rhizoid or filamentous? Any question about that? You can look at the colony elevation, and there are flat colonies, raised colonies, convex colonies, pulvinate colonies, which are super convex colonies, and then umbunate. I'll never ask you to distinguish between is the colony convex or pulvinate. The size of the colony can be punctiform. That's about the size of the tip of the pen. It can be small. That means it's smaller than the head of an eraser. And this pen has a very small edge. So let me take a look at another pen. So small is smaller than the head of an eraser or the head of this pen. A moderate is about the size of the head of the pen. And then large is larger than the head of the pen. Colonies can be smooth or rough. They can be glistening or dull. They can be non-pigmented or pigmented. And they can be opaque, translucent, or transparent. Very few bacterial colonies are transparent, and those are actually difficult to see. Some of them are translucent, but the vast majority of colonies are opaque. So when we look at these here, we can see that that is an opaque colony, and it's round. It is opaque, shiny, okay, and it is elevated. It's probably convex. But to see that specifically, you need to take off the lid of the plate. Take off the lid of the plate, and then look at the colonies at an angle, and then you can easily tell what the elevation of the colony is. It's shiny. Oh, and it's pigmented. If a colony is pigmented, it has color. So any colony that's white, off-white, or gray is non-pigmented. But this one looks yellow, so does that one. So those are pigmented colonies. Any question about that? This would not be a pigmented colony because black is not one of the colors, meaning you need a color other than white and black to be colored, to be pigmented. Now the good thing is, if I ask you about the colony characteristics, I will give you this table, but you have to be able to use the table and interpret the table. All right, when we're looking at a plate, Actually, let's move on because we're a little behind schedule. We can count the number of colonies, and that is the abundance of colonies on the plate. There's too many to count here, but it's something like 30 to 50 colonies on this plate. And that would be the abundance. The diversity is the number of different kinds of colonies on the plate. Now, I can distinguish that this colony is different than that colony, so that makes 2. And that colony is different than these, so that makes 3. These 3 are possibly all the same. That one may be the same as that one, but that one has a different color and shape than all of these, so that makes, what do we got, 4 now? And then this one is a different color, and that one's a different color, and that's kind of pinkish, so that's a different color. That one might be the same as this one, it's probably different. Anyone count how many I have now? I sort of lost my count. I was 4, 5, 6, 7. So there's at least 7 different colonies on this plate. So the diversity is at least 7. And if you get anything near 7, and it might be as high as 12, like these are really small colonies, that might not be the same as this one. So that would be 8. Let me look. That one could be the same as that one. So let's say it's between 7 and 10 for the different types of colonies on this plate. As long as you're close to something like 7 to 10, I will okay it. Like if you say 5, you're a little bit low, but I'm not worried about it. But if you say 2, I'd say, that's wrong. There's clearly more than 2 different colonies on this plate. And then if you say there's 50 colonies on the plate, and you say there's 40 different ones, I'd say, no way, there aren't that many different, because this one and that one and that one, 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 and that one are clearly all the same colony. And then this one, and that one are also the same, and that one, the same species. Okay? So you have to be within a range to get the right answer. And generally, it's not that difficult. Obviously, that one's kind of difficult because it's got so many colonies on it. This one's a little better because all of these little yellow ones are clearly the same species, and there's a bunch of yellow ones here. That yellow one is different, so that's 2, and then 3, and that might be different, so that might be 4. And there's a, I don't know, a grayish one, so that's 5. And there's an off-white one, so that would be 6. And that could be different than that one, because it looks a little wider, but it might be just because it's smaller. So something like 6 to 8 would be the diversity on this plate. Okay? Any question about that? And as long as you're close, I will let it go. But if you're way off, like I said, if I'm counting 6 and you count 2, that's going to be wrong. And if you count, oh, there's 20 different ones, that's going to be clearly wrong too, because, well, there's clearly only 30 to 40 colonies here, and it's impossible to count these right here and those right there. Any question about any of that? A better example is the abundance in this picture of cats is there's 7. 1, 2, 3, 4, 5, 6, 7. And the diversity is there's 2. There's lions over here and cheetahs over here. You do need to know the terms abundance and diversity. And for some reason, some students get the two terms mixed up, and then they're obviously wrong. So know the terms in this lab, and then go through the, you don't need to do the references. Go through and do the lab, and let me open up the worksheet. Write your answers in the worksheet, and then submit it in the worksheet place. Let me see if I can find that. So click on this link here, and then you should be able to upload worksheet 1, and you should be able to find worksheet 1 in here. And I believe it's the second link there. There's the worksheet 1. And then just upload it. You'll have, instead of a speed grader, you'll have a link for uploading the file. And do that by 11:59 p.m. Saturday, because that's when it's due. And together, these two labs are worth 5 points, so each one is worth 2.5 points. All other labs will be worth 5 points. Let me go back to the worksheet. So read the worksheet and then answer the question. You do not need to answer procedure 1, what's in the air you breathe. That is just something for the worksheet to make it, I don't know, stick out, and that is when you're breathing things in, you're breathing microbes in. So this plate was exposed to the air and then incubated for 24 to 48 hours. And it's kind of curious, we don't say how long it was exposed to the air. And then we're asking you, what's the colony abundance? You just need to count the colonies. And then tell me what the colony diversity is on this plate. And then also tell me what you would do if you're working with the plate and you're, I don't know, streaking it or working with it. What would you do to reduce airborne contamination of the plate that you're inoculating? And it's quite simple, actually, when you think about it. Procedure 2, go ahead and read through it. What's on your arms when you put your arms on the bench? So I assume it's what is on the bench. And how effective table disinfection is. Answer this question. There's the before and the after. And then it asks you, what's the purpose of allowing the table disinfectant to air dry when you put it on the table and then spread it around? Okay? And then do procedure 3. We're looking at what's on your hands. And you're washing your hands with soap and water. This is the hands before washing them, and this is the left hand after you wash them. And then answer the questions. Does hand washing work? And then suggest one reason why hand washing did not result in complete removal of microbes. And there's a hint there you can read. Procedure 4, when you kiss your significant other, exactly what are you giving him or her? You don't need to answer that question. It's just starting the procedure. And what we did was somebody kissed an agar plate here, touching the lips there, and then somebody put their tongue on the agar plate there. And you should note that I put a note that you guys should read before you answer the questions for the tongue plate. And the question is 1A, what plate has higher colony abundance, the lips or the tongue? And B, why do you think this is the case, even though your lips and the tongue are very close to each other? That is, what might cause a greater colony abundance on the lips or on the tongue compared to the other. And then lastly, we mentioned that when we were incubating these plates, we incubated the tongue plate at a higher temperature than the lip plate. And why did we do that? So answer that question. And then lastly, are there microorganisms in the soil? And here we inoculated a plate with a little bit of soil. And you need to tell us, what is the colony abundance? And is it even possible to accurately answer this question? Explain your answer. And is there greater abundance of colonies in the soil compared to the lip plate? So write up your answers and then submit them in Canvas. I will be here until 8:30 to help you in the lab. So if you have any questions, just ask. If you don't see me here, it means I've stepped away from my desk. I've probably gone to get a bite to eat or something, but I will return in no longer than 2 minutes, and then you can ask your questions. All right, get to it. If there's any questions, you should ask it now.